Hi everyone, my name is Alyssa. I am coming from Vermont, which looked like this a couple weeks ago. And I'm excited to talk to you today about gene regulatory analysis in leukemia. So first I want to emphasize with how expensive gene... Okay. I want to first emphasize how important gene regulation is. Gene expression is highly regulated by different layers of epigenetic modifications like DNA methylation or histone modifications. And ultimately, these modifications culminate into changing the chromatin accessibility state of the cell, where genes with regulatory regions that are open and accessible for transcriptional machinery are expressed, and genes with compacted chromatin are repressed. And transcription factors are really important in regulating the accessibility of genes. In particular, lineage-specific transcription factors, which are active in just the lineage that they are pushing the cells in, are important to express the genes that are specific to that lineage and repress genes from other lineages. And this is how we get one progenitor cell that becomes many cells of different fates that all stem from the same DNA. Lineage-specific transcription factors are especially important for B cell development, where the actual stages of B cell development are partially defined by the transcription factors that are active at that time. I'm showing here just a few of those. Ikaros in the top and PAX5 in the bottom are actually expressed and active throughout B cell development. These transcription factors are also commonly mutated in cancers. So in cancers of developing B cells or acute lymphoblastic leukemia, some of the top two mutations are in these two genes, IXDF1, which encodes Ikaros, and PAX5. The common mutation that patients have when they have an Ikaros deletion or an Ikaros mutation is actually a deletion of the exons containing the DNA binding domain of the protein, and this renders the protein a dominant negative and non-functional. And patients with this deletion have a worse overall survival and increased chance of relapse. And so from this, Ikaros was named a tumor suppressor. However, we're not fully sure how it's doing its tumor suppressor function. And so my work is to understand how Ikaros is working to change the gene regulatory programs in these cells, and ultimately understanding the altered gene regulation will reveal oncogenic transcription factors and pathways that could represent therapeutic possibilities. So in order to do this, to understand the gene regulatory environment, we have a number of techniques to use. First is ChIP-seq, which got a little introduction earlier, so I'll just go through this quickly. But we can detect the genomic regions of transcription factor binding using an antibody targeted to our transcription factor of interest, and then pull out that antibody and with it the target gene or target transcription factor in the DNA that it was bound to at that time. And we can sequence that DNA and identify regions where that transcription factor is throughout the genome. We also use the assay for ATAC-seq, the assay for transposase accessible chromatin, followed by sequencing. And this utilizes an enzyme that we add on to the cells, and it cuts the DNA wherever it can fit into the chromatin. And so from this, we can purify the cut fragments and sequence them. And the reads correspond to regions of the genomes that are open, chromatin is accessible, and genes with open chromatin are transcriptionally accessible. Okay, to model Ikaros tumor suppression, we use two cell lines that are from patients with B-cell leukemia. And these, these cells harbor the dominant negative deletion in the Ikaros gene. These cells are stably transduced with vector where we can reintroduce wild-type Ikaros or control. And so we're basically rescuing wild-type Ikaros function. And we see when we check protein levels after adding our inducing agent to get expression, we get higher levels of protein of our wild-type Ikaros compared to control. And most importantly, what we see when looking at cell cycle analysis is that upon Ikaros induction, the cells exit the cell cycle and enter G1 phase. And so this is really important. It confirms that our model is working to show tumor suppression. So to understand the altered gene regulatory environment, we first started by looking at global gene expression using RNA sequencing. And we see many genes that are both increased and decreased in expression upon Ikaros induction. We also did perform ChIP-seq to understand DNA binding locations of Ikaros. And when we compare those two, we see that Ikaros has binding sites at approximately a third of the differentially expressed genes, and it's not skewed towards either upregulated or downregulated genes. So this indicates that Ikaros is directly regulating these genes. To look at what these genes are involved in biologically, we can see that they're enriched in pathways related to cancer, like the MYC pathway here, which is a classic oncogene, and also the BCR signaling pathway, which is really important in B-cell development and is commonly altered in B-cell leukemia. We next looked at the chromatin accessibility landscape in these cells, and again, this is globally altered. We see regions first that are unchanged between conditions in both cell lines, and then we see regions that lose a little bit of signal upon Ikaros induction, and also regions that gain signal. This differential accessibility is easier seen in this plot to the right, where each dot represents a region. And we have regions that change that have increased accessibility, a positive log fold change, and decreased accessibility. And actually about 2/3 of differentially accessible regions are losing accessibility. Comparing this to our regions of Ikaros binding, we see that really most of the overlaps between differentially accessible regions and Ikaros binding are among regions that lose accessibility. And this is consistent with reports in the literature of Ikaros being a transcriptional repressor. So to put this into context for you, here's an example at the gene CD34, which is a gene involved in stemness. So less differentiated, more stem cell-like genes have higher expression of CD34. And what we see when looking at our Ikaros ChIP-seq is that we have Ikaros binding signal at this transcript, and indicating that Ikaros is directly bound to this gene or in regulating this gene. Looking at accessibility signal, we have high accessibility in our control cells, and then upon Ikaros induction, we lose that accessibility in blue there. And looking at gene expression as well, expression of CD34 is repressed down to almost 0 counts. So far, we've been looking at genes that are directly regulated by Ikaros with Ikaros binding sites present, but that only accounts for a third of the differentially expressed genes. So to uncover the rest of this picture, we can use a number of tools. First of all, upstream regulator analysis is something that takes the whole input of differential gene accessibility and predicts that transcription factors that might be responsible for the changes seen. And so what we see, first of all, most importantly, our most significant pathway that's activated is ICZF1 or Ikaros. And this is important to see, first of all, as a sanity check, because this is the perturbation that we're adding back into the system. Second of all, the next upstream regulator that sticks out to me, the second most significant one, is actually MYC again. Again, that's a very prominent oncogene. And so we're working in the lab to further investigate this connection. We can also do motif analysis and look at differential motif availability within chromatin accessibility peaks. So again, if motifs are either more enriched in ATAC peaks in one condition versus the other. And then we can put this all together using paired expression and chromatin accessibility, or it's also called PICA analysis. And it creates networks that identify condition-specific networks. So here I'm showing the networks that are specific to each condition. And notably, what we see in our control cells, so these are the cells with the Ikaros deletion, the top interactions between transcription factors, the large ones, and target genes are transcription factors that have been known to be oncogenic in other cancers. Whereas comparing this to our gene regulatory network that's specific in our Ikaros-induced cells, we see a number of transcription factors that are present in normal B cell development and present in mature B cells. So overall, I've used a systematic analysis to characterize the epigenetic landscape in B-ALL and its deregulation upon Ikaros deletion. Ikaros binding sites are enriched at TSSs of genes related to cancer and BCR signaling and at genomic regions that lose accessibility. And we can look at the indirect effects of Ikaros by looking at the altered gene regulatory networks in the cells. And ultimately, we're hoping to identify oncogenic transcription factors or pathways that may represent therapeutic vulnerabilities. And with that, I would like to thank everyone in my lab and also the organization for letting me talk today. And I can take any questions. Thank you.
Okay. I was wondering in your last network if the genes that are regulated are up or down.
If they're up, yeah. So we have that information. So they're both upregulated, they're both upregulated and downregulated. However, we don't know, well, we can't represent on this network whether the genes that are upregulated, they might be negative regulators too. So yes, it kind of goes in all directions for every